All right, hello, my name is Chase Baker.
Golzar Saranj.
Nicole Pierce.
Sophia Velasco.
Chastity Mendez.
And this is our presentation on the gut-brain connection and disease. So today we're going to be talking about what is the microbiome and nosocomial infections, bacterial influence on eating disorders, the evaluation of gut microbiome in patients with major depressive disorder, also known as MDD, microbiome and Alzheimer's, and the gut microbiome in multiple sclerosis. So what is the microbiome? Picture a bustling city on a weekday morning, the sidewalks flooded with people rushing to get to work or appointments. Now imagine this at a microscopic level, and you have an idea of what the microbiome looks like inside our bodies. Consisting of trillions of microorganisms, microbiota, microbiome, thousands of different species. These include not only bacteria, but fungi, parasites, and viruses. In a healthy person, these bugs coexist peacefully, with the largest numbers found in the small and large intestine, but also throughout the body. The microbiome is even labeled a supporting organ because it plays so many key roles in promoting the smooth daily operations of the human body. So how does the microbiome play this role? Microbiota stimulate the immune system, break down potentially toxic food compounds, synthesize certain vitamins and amino acids, including the B vitamins, vitamin K, and the infamous neurotransmitter serotonin. For example, the key enzymes needed for vitamin B12 are only found in bacteria, not in plants and animals. The microbiota of a healthy person will also provide protection from pathogenic organisms that enter the body, such as through drinking or eating contaminated food or water. So now we're moving to nosocomial infections. Nosocomial infection is contracted because of an infection or toxin that exists in a certain location, such as hospitals, which is most likely the place people get them. People now use nosocomial infections interchangeably with healthcare-associated infections or hospital-acquired infections, HAIs. In American hospitals alone, the Centers for Disease Control estimate that the HAIs account for an estimated 1.7 million infections and 99,000 associated deaths each year. The prevalence of CDI, which is C. difficile infection in U.S. hospitals, is estimated to be 13 of 1,000 patients. Approximately 75% of cases are hospital acquired, and resulting in healthcare expenditures of US dollars to $9,000 to $15,000 per patient, or an estimated 1.5 to 3.2 billion annually. 20% of the people that are treated with antibiotics will develop recurring infections. And that was the old way of treating this. The new way of treating this is with fecal transplants. This is also known as bacteriotherapy. Fecal transplants is the transfer of stool from a healthy donor into the gastrointestinal tract for the purpose of treating reoccurring C. difficile colitis, also known as CDI. The cure rate with this fecal transplant is 80 to 90%. So a surprising finding looking at GLP-1, glucagon-like peptide, it's a gut hormone that is mediated by gut microbiome, increased in a handful of patients reporting weight loss. And they suspect that it's GLP-1. They cannot prove this yet, but this is what they mainly suspect is the responsible. And they also had the opposite effect. Some people gained weight after getting a fecal transplant, and we haven't looked into that either. So mice and rats are really great model organisms to study the microbiome. They have a much more diverse microbiome than humans, but nonetheless, you can breed sterile rats that will rule out any variables of external microorganisms playing roles into the nervous system. So, for instance, there has recently been shown that fecal transplants from a healthy donor restored both gut microbiota composition and function in human patients. This is what we know, and this has been used to treat people with C. diff infections. And so, looking at a study with these mice, you have lean mice and fat mice that you can create from sterile mice just by feeding them the fecal transplants of fat mice or lean mice, and that's how you can alter their microbiome. And looking at GLP-1, which acts as a neurotransmitter on the vagus nerve that talks to the brain, looking at this diagram here on the bottom right of the graph, they're still trying to figure out how the bacteria are influencing GLP-1 to signal to the appetite change and nervous system inflammation, which has been seen to treat IBS and a bunch of different other diseases, but specifically C. diff. We're seeing people lose weight, and we're seeing the same with these mice. And we are studies away from finding out this mechanism.
So I did my research on the gut affecting those with eating disorders. The eating behavior is mediated by a network of interacting neural circuits in the brain, such as the hypothalamus. When someone's hungry, the human biology compels them to seek out food. Once you eat, you usually feel the reward of pleasure. If you continue eating, you usually reach a point of satisfaction. Some, though, are able to override this hypothalamic balance signal. This results in the inability for one to overeat despite reaching a comfortable level of energy storage. You can also undereat despite needing immediate and stored energy. Researchers question if there was a protein that is or could be responsible for people who suffer from EDs. Next slide. So the French National Research Institute and the University of Rouen claim to have discovered some bacteria in the gut that may interfere with the way the body regulates appetite, at least in rodents. Next slide. They found that a protein called CLPB, produced by the gut bacteria E. coli, mimics the regulatory hormone melanotropin. When the immune system recognizes this protein, it makes antibodies to attack it. The problem is that CLPB is almost identical to melanotropin, which does work to regulate the appetite. When antibodies attack the protein, they also attach onto the hormone, so the body doesn't understand when it's satisfied. In anorexics, this process makes them feel fulfilled too early. In the case of bulimia, people tend to overeat since they don't feel full until it's too late. Next slide. So they used mice for this study. They had two groups of rodents. One was given strains of modified E. coli not producing CLPB, and the other received a common gut bacteria. They found that the first group did not change their eating behavior and levels of antibodies, whereas the second one did. They were also able to test their hypothesis on 60 test subjects and noticed that those who had a high antibodies also were the ones suffering from the most severe eating disorders. It was found that these patients had high plasma levels of antibodies to CLPB protein and melanotropin. These findings confirm the involvement of the bacterial protein CLPB regulating appetite. So as you can see in this figure on the right-hand graph, it's the presence of CLPB in the plasma of a human, and you can see that ED stands for eating disorders, and the ED compared to the control, you can definitely see an elevated level of CLPB, and in comparison to the chart on the left, you can see the levels in rats, so they're both pretty similar. They're both elevated. Next slide. Next slide. This diagram shows the mechanism on how anorexia nervosa is cycled through the body. So the beginning on the left-hand side of the circle, you can see that the dietary restriction will cause an increase of enterobacteria. That will cause a prolonged stationary phase, which will in turn increase the levels of the CLPB. So therefore, the permeability will increase, and the CLPB protein can activate anorexigenic neuronal populations, such as POMC, whose response will lead to an increase in satiety. And by understanding this physiological factor, the aim would be to try and neutralize the protein using specific antibodies without affecting the appetite hormone to potentially help restore a healthy food intake.
Hello, I'm going to talk about the altered gut microbiome in patients with major depressive disorder, or MDD. The first class of bacteria I'm going to discuss is Prevotalaceae. Chen and Jiang observed in their respective studies lower levels of this bacteria in patients with MDD compared to controls. This bacteria produces bacterial metabolites called short-chain fatty acids, or SCFAs. SCFAs play a key role in neuroimmunoendocrine regulation. One thing they can do is cross the intestinal barrier into the bloodstream, cross the blood-brain barrier, and it can modulate brain functions that include influencing behavior, affecting neural signaling, and altering the production of certain neurotransmitters. When SCFA production is altered by Prevotalaceae prevalence, dysregulation in this modulatory system can occur. Researchers Ozawa and Liu also found decreased levels of bifidobacterium in patients with MDD compared to controls. Increased levels have been associated with positive mood. Yang and associates observed the intake of bifidobacterium in rats may prevent the onset of MDD from stress. This may mean that lower levels of bifidobacterium may result in the onset of MDD from stress. Next slide. Wang, Zhang, Liu, and Jiang also noted decreases levels of Faecalibacterium in patients with MDD compared to controls. A negative correlation has been observed between this bacteria and depressive symptoms, but the underlying mechanisms connecting them remain unclear. Despite these findings, there are some major limitations in the current studies of human gut microbiota in mood disorders. Specific gut microbiome linked to gut variation among studies is likely due to study populations, methodology, and the presence of confounding factors such as diet, age, sex, and lifestyle. Next.
So I'll be addressing the microbiome and its association to Alzheimer's disease. So before we get into it, we'll have to discuss what APP genes are. And APP genes, they make amyloid precursor proteins, which constitute for the amyloid plaques, which are also known as the misfolded proteins that are found in the brains of Alzheimer's patients. So the photo to the right shows a side-by-side comparison of a normal brain to a patient with Alzheimer's, and there's also a close-up of the neurofibril tangles as well as the amyloid plaques. So next slide. So scientists speculate, oh, I think you went too far. So scientists speculate that the dysbiosis, which is a microbial imbalance or maladaptation, could have a role in the setting production of these amyloid plaques in patients with Alzheimer's. And so Hirsch and a team of scientists set out to understand the effects of the microbiome. They compared the presence of microbial dysbiosis in gut levels in APP transgenic mice to wild-type mice, and they then assessed that the colonization of germ-free APP transgenic with the gut of the microbiome of APP transgenic mice were able to enhance amyloid B-related cerebral damage. However, the gut microbiota from the wild-type mice had little effect on amyloid beta levels within the brain. You can go to the next slide. So this photo shows the results of those findings. And it's a cross-section of the cortex and the hippocampus within these germ-free mice and the conVR mice. And these two pictures are taken at 3.5-month-old mice and an 8-month-old mice. And there's a huge distinction between the amyloid plaques, which could be seen by those little bright markers of fluorescence. And so these essentially these pictures demonstrate that the amyloid beta deposits are reduced in the absence of gut microbiota in the germ-free mice. Next. So lastly, the study also found that levels of Akkermansia were decreased in APP transgenic mice, and Akkermansia is a gut bacteria that increases the gut barrier integrity. And so the depleted levels of this bacteria can essentially lead to a disrupted gut barrier, which can also cause an influx of inflammatory components, which will essentially in turn contribute to more dysbiosis. Next.
So I did my research on the link between gut microbiome and multiple sclerosis. Next. So multiple sclerosis is described as a potential disabling disease of the brain and spinal cord or the central nervous system. The immune system attacks the protective sheath that covers nerve fibers and causes communication problems between the brain and the rest of the body. MS can eventually cause permanent damage or deterioration of the nerves. The cause of MS is unknown. The signs or symptoms of MS vary widely depending on the amount of nerve damage and which nerves are affected. The most common symptoms affect movement, such as numbness or weakness in limbs, tremor, lack of coordination, unsteady gait or electric shock sensations with certain neck movements like bending the neck forward. Other symptoms include slurred speech, fatigue, dizziness, tingling or pain in the body, problems with sexual bowel and or bladder function, blurry vision, prolonged double vision, partial or complete loss of vision with pain during eye movement, usually in one eye at a time. Some who suffer from MS experience long periods of remission, while others experience severe symptoms such as losing the ability to walk independently. Treatments include immunosuppressive drugs to inhibit or prevent this activity from the immune system, corticosteroids to reduce nerve inflammation, chemotherapy drugs to kill white blood cells, acupuncture to provide relief from some MS symptoms, and physical therapy for stretching or strengthening exercises to ease performance of daily tasks, manage leg weaknesses, and other gait issues. Many of these treatments, acupuncture and physical therapy excluded, may halt or slow the progression of MS, but also suppress or weaken the immune system with very serious, severe, and unpleasant side effects. Next. Myelin is like the insulation coating on electrical wires. Once the myelin is damaged and the nerve fiber is exposed, messages that travel along the nerve may be slowed or blocked, or the nerve becomes damaged itself. Most people with MS have relapsing remitting course of the disease. They experience new symptoms or relapses of symptoms for days or weeks and then improve. But 60 to 70% of these MS patients eventually develop secondary progressive MS, a steady progression of symptoms with or without remission periods. Primary progressive MS is steady progression of symptoms without any remission. The MS patients examined by the University of Zurich Clinical Research Priority Program found gut microbiota may have a bigger part in the pathogenesis of the disease than previously believed. T cells are the immune cells responsible for pathological processes and react to the GDP-L-fucose synthase protein. Next. To translate this finding into therapy, the pathological autoreactive immune cells are significantly targeted. Blood is drawn from the MS patients, and immunoactive protein fragments are attached onto the surface of red blood cells in a laboratory. The treated blood is then reintroduced into the patient's bloodstream. The belief is the attached fragments will help to re-educate the immune system, making it tolerate its own brain tissue. This therapy aims for effective targeted treatment without severe side effects. Next. Thank you for listening to our presentation.